Welcome back to the Al-Itaq Conference. I'm Dr. Kate McLachlan, the moderator for this session, The Promise of Genetic Research, which is going to focus on what genetics has to offer the Arabian horse community. It is my pleasure to welcome our speaker, Professor Samantha Brooks from the University of Florida. Professor Brooks has a doctorate in veterinary science, specializing in equine genetics. She has published research on a range of topics, including health issues, genetic diversity within the Arabian breed, and the influence of Eastern stallions in the development of horse breeds around the world. We are very grateful to have her here today to share her knowledge and insights on equine genetics. Her talk today, History Written in the DNA, Genomics for the Arabian Horse, will look at what we can learn about the history of the Arabian from its genes. I'm pleased to hand the floor to Professor Brooks to start her talk. If you have questions, please post them in the chat, and we'll get to them at the end when we will have time to answer them. Over to you, Professor Brooks.
Excellent. Thank you, Kate, for that introduction. And thank you to our hosts and organizers today for putting together this lovely program. I'll just share my slides with you now. And hopefully technology will work as planned, and everyone can see my slides now, yes? Excellent. Well, I'm really excited to speak on this topic today. It is an exciting time to work in genetics and genomics with the new developments in technology that have arisen for human medical genetics primarily. Our other species of importance, especially the horse, have also benefited and can now apply the same technologies in their own setting. As a result, the past 10, 15 years or so have seen a number of remarkable discoveries and new in-depth descriptions of the ancestry of horses in general, but especially the Arabian horse. I'm excited today to share with you just a few illustrations of what the recent science has brought to light with regards to ancestry of these remarkable animals. I do apologize. I have many things I would love to share with you, but I'm only going to bring a few vignettes of some of the recent work. I'd like to start with some basics. I think it helps us to review what I mean when I'm talking about genetics and DNA. It's been a while since many of us were in school. When I speak about genetics, I'm describing the instructions for life that are written in the DNA sequence, which is stored within the nucleus of nearly every cell. That DNA is comprised of individual nucleotide bases that are strung together almost like pearls on a string. The changes in the sequence of those individual nucleotides, which they come in 4 varieties, and we abbreviate those as A, C, G, and T. And the changes in those sequence or that text underlie the differences that occur between species and between individuals. In the horse, that sequence is very similar in length to what we find in humans, about 2.7 billion individual nucleotides. So you can imagine this is like a very large set of encyclopedias. It could easily fill about 3 full-size bookshelves from floor to ceiling if written out as text in a font similar to what you see in an old encyclopedia set. For both horses and humans, the genome contains around 20,000 functional genes. And genes, you can think of those sort of like figures or within those encyclopedias. The genes provide a very explicit set of instructions for creating proteins. The great thing about genes is that they follow a very strict and easily understood system of coding. They write into code based on a triplet set of nucleotides, the correct amino acid to put together to create a protein. It's just a set of instructions for creating the machinery that the cell needs to produce many of its vital functions. That means, unfortunately, that if we can understand this section very well, we understand protein coding genes very well, they comprise just 3% of the entire genome. That means the other 97% is written often in a code that is far too complex for us to perfectly understand today. So there's a lot of work left to be done in genetics and in genomics. Now, any gene and the sequences between genes can have variations, and variations will be our tour guide today because they help us to describe the differences between individuals, both in their appearance, their phenotype, and also in their relatedness. So two individuals who are closely related often derived their genomes from the same ancestors and frequently contain many of the same patterns of variation. Variation provides for us, it's kind of like a barcode that helps to tag the origins of that genome through the passing of many generations. Oh. Well, that's going to be, I may have a technical difficulty with my slides there, but just to show you, in real life, in the lab, when we work with DNA, this is what it looks like. So this, the cottony structure that you can see just floating within this tube of alcohol in the laboratory, that is the DNA from about 10 mils of horse blood, from one sample from one horse. So when we get to the lab, we strip away the membranes and the proteins that package the DNA, leaving behind just that molecule comprised of strings of nucleotides. And once we sort of dry it out in alcohol, it condenses into a structure that is very visible and really quite beautiful. It's an exciting moment for us in the lab because much of what we do today is on the computer. But what you need to know, virtually everything you need to know, I should say, to create a horse is written within that fluffy white string inside the tube. Now, as I mentioned, what is benefiting much of science, especially in genomics right now, is our shifts in technologies. So not that many years ago, when I learned to drive, if I needed to get somewhere, I would need to use a paper map, maybe do some math based on distances and what speed I expected to travel. And I would probably use an analog watch to determine my time of departure and track my progress. These days, the advances in electronics have meant that all these functions and so many more are now packaged automatically within our cell phones, which we now seem to carry with us everywhere we go. With the science of genetics and genomics, the progress has been no less spectacular. So previously, it would have taken me 4 to 6 weeks to generate just a small amount of sequence covering perhaps 1,000 or 2,000 nucleotides of a gene that we might be interested in. And that was if we knew where the gene was, because we didn't necessarily have a full genome map. To today, we have very inexpensively technologies like what we call a SNP chip. So SNP stands for a single nucleotide polymorphism. That's just a type of variation. And these chips are the size of a microscope slide, just 1 inch by 3 inches. And in this case, this particular model that I show you here in gray and black, it holds 12 potential testing lanes on top of that slide, or room to test 12 horses. Each one of these lanes can determine the sequence at around 100,000 genetic markers. So what would have taken 4 to 6 weeks previously can now be accomplished in just a few hours, especially if we think so far forward as some of our genome sequencing technologies. As a result, we can create maps of genetic variation very quickly and with unprecedented levels of detail. So, with our background under our belts, I'd like to think a little bit about how these technologies illuminate the history of the Arabian horse as it is written within their own genome sequences. I think that when we consider concepts of history and breed development, we have to sort of take a moment and think about what is a breed, because often no two humans can agree on a definition of many things. But when it comes to the idea of what is a breed or even what color a horse is, often two horse people will have independent opinions. Biologically speaking, we do have some general guidelines, though. And I'm giving for you an example here of the family tree of the dog. When we look at each of these dogs, we can, in our modern breeds of dog, we can divide them into general types, and those types frequently correspond to their geographic origin. That is a result of a very logical effect that dog families and who you breed to are often directly correlated to the distance between the two dogs, at least historically. Today, of course, in the horse, with the use of cooled and frozen shipped semen, our horse populations have developed the ability to create global breeds that reflect relatively little geographic stratification. And indeed, in history, because the horse has been such an icon of human transportation, they often defy geographic stratification just by the ability to move along with human populations. But in any case, we can find categories and types that correspond to the genetic variations found within the genome. This cartoon of these little pie graphs actually illustrates the genetic variation that is found in the domestic dog. By reading those individual barcodes and grouping them into colors based on the commonalities, where the barcodes are similar and where they're dissimilar. And in this case, each color also happened to correspond to an ancestral group, either from a geographic distinction or potentially from a type distinction. So if you look at two dog breeds, let's say the Basenji and the Rottweiler, you can see that both of those breeds share the dark gray ancestry, but the Rottweiler uniquely possesses the red ancestry. Now, one of the advantages of this type of analysis is that it's done entirely in the computer, and it's based on the sequences of the genomes themselves and where they are similar. It is not based on human definitions of the name of a breed or the history of a breed or on written pedigrees. So, finding the correlations between what the red ancestry is and something that is human readable with regards to history is often very difficult. But letting the computer find those biologically significant ancestral trends without the influence of human opinions nonetheless gives us an objective opportunity to discard prior assumptions and discover new phenomenon and event as we without the biases that come along with being human. So we have done a very similar exercise in horses. This is from 2008 and was really quite illustrative. So done in the very same method, we have these pie graphs for several global breeds or examples. And right away, you can see the difference between the horses and the dogs. Each individual horse breed possesses many different colors as compared to the dog breeds. This is due to that effect I described earlier. Even earlier in history, prior to the use of shipped semen or traveling stallions, the horse itself traveled with human populations virtually around the globe. Horses are also migratory in nature, perhaps more so than most dogs or dog ancestors. So they traveled and shared their genomic variation more broadly. It's a unique difference between the species. Additionally, though, as we have developed these breeds, we have used different geographic or ancestral populations to attempt to improve those breeds and change some traits of interest. Most notably, of course, the use of the Arabian horse to improve the athleticism and refinement of various breeds. And so you can see that modern admixture contributes to this effect as well. We can also group our modern horse breeds globally into something of a family tree based on the similarity in those genetic variations. And so in this follow-up work, these authors looked at genetic variations across small populations of each breed. There were about 20 horses in each breed category. And then based on the number of similarities, created this distance tree. So in the blue circle, you can see a grouping of what I'll call sort of the general light horses. That includes thoroughbreds, our sport horses and warm bloods, as well as the stock horses. And you can also appreciate in the yellow circle, we have our Lusitanos, Andalusians, Paso Finos, and horse breeds that come from, in general, sort of a Hispanic or Iberian ancestry. And there on the bottom in the red circle, you see our sort of our Oriental and Middle Eastern group, I will say. It includes the Arabian and Al-Itaq and the Caspian. Other breeds might fall intermediate to these branches, in part because they may be a composite of the two types. So we know that the Arabian has contributed to several of our North American breeds, like the saddlebred, as has the thoroughbred. So they sort of fall in between one another. And then our cold blood horses, as we call them, the Norwegian Fjord, Icelandic, falls on their own branch, as do many of our hardy ponies. The key thing here is that the length of the branches on this family tree correspond to the distance between those breeds. So not only are they located in clusters, but if they are on very short branches, they're very closely related to one another. We've just translated the number of differences into a geometry of this stick and branch structure. Now, we have looked at the ancestry of the Arabian horse using genetic variation in a couple of interesting papers, and I'm actually going to go back to one of our early ones just for a moment to help to better understand the concept. So here we have a lovely example horse. If we were to use the genomic structure, the variety of genetic variation in this horse's genome to catalog his ancestry relative to a library or a reference population, we can then divide his genome up into the pieces that are derived from each of those ancestral populations. So in this case, we are representing relatedness or similar genetic variation, not by sticks and branches on a tree, but by colors. So if we knew from our reference population that there were groups of horses that had trends or tended to share variation as types, we can then read his genome and assign to it a proportion that likely came from each of those ancestral populations. So this horse, we might say that about 50% of his ancestry belongs to the blue group, 25% to the yellow group, and 25% to the red group. And again, because the computer does this comparison without the prior assumptions provided by humans, the definition of what the blue group is, what the yellow group is, and what the red group is requires further study. And it shifts and changes depending on the reference library of horses to whom we compare him. That's actually a fantastic feature of this particular technique, because we can learn one part of the story by comparing him to global populations of the horse. And we can learn a different part of the story by, say, comparing him to his nearest relatives. So in this example, this paper was one of the first works to examine the ancestry of the Arabian horse. And we had just a small population of about 50 animals, but it gives us a very nice little illustration. In this set of 50 animals, we could readily identify some commonalities or types. So among our samples that were labeled as Egyptian in origin, they frequently had this strong red type. And among those who were Polish, we saw the yellow pattern of genetic variation. And as you might expect, when we looked at horses who just lived here in the United States and often had diverse and admixed ancestries, at least in terms of Arabian subgroups, they had a pattern of mixture showing that they had benefited from both the Polish yellow ancestry and from the red or Egyptian type ancestry. Now, these patterns, they do shift and change based on the reference population slightly, and there is always some level of commonality that predates the diversity of these subgroups. For example, these are all horses, and they are all Arabian horses. And so in some cases, the variation, the divergence that we see is due to chance, and in others, it may be sources of variation that actually predate the divergence of the subgroups. But in general, this method does a really fantastic job of describing overall trends. In a follow-up paper, this group used this similar technique to look at the common ancestries among the modern breeds of horse from a global perspective. So in this plot, just like in the previous plot here, each column, each vertical column is one horse, which is fairly easy to see when we have a small number of horses. When we have a large number of horses, it becomes really quite blurred. I believe the study had over 300, but I would have to go and look up the exact number. As you can see, even in this zoomed out version of the plot, the computer has assigned to each breed a pattern of variation and coded it in as a unique color. So the Clydesdale you can see on the left, the Clydesdale signature of genetic variation is now light blue. And the Shetland pony on the far right, you can see is now quite orange. The Arabian horse here with their unique breed signal is shaded in pink. Now, this plot has multiple rows to it because you can consider these commonalities at different level of detail. It's like a microscope or a telescope. You can zoom in very close, and you also can zoom out and get a much wider angled view. So the smaller numbered rows at the top are the wider angle, and at the bottom we have the most detailed, the finest level of view. That gives us some additional power to examine the story from different angles. So if we look more closely at some individual populations at the most detailed level, you see that the American paint and quarter horse tell a story that agrees fairly well with our written history, and that we know that they are admixed. They have strong green and strong yellow signals coming from the populations from which they were derived. The Al-Itaq is a little bit more homogeneous, showing that strong chocolate signature of variation. Here are some standard breds. The interesting thing here is that they're considered largely the same breed in the same registry, but within the breed, we have notable differences in that in Norway, they have a strong light green signal, but here in the United States, they now have a predominant dark gray signal as well. If we look at the thoroughbred, both from the United Kingdom and from the United States, they're the source of that strong lemon yellow pattern of genetic variation, and that agrees quite well with what we see in the paint and quarter horse because we know the thoroughbred was used quite a bit to create those breeds. And if we examine the Arabian, the long history of careful guarded breeding has produced a strong pink identity that is quite characteristic of that particular breed. Now, we sought to apply the same approaches, but to focus strictly on the Arabian breed itself. So we collected a large sample of over 700 horses, and we worked as much as possible to gather diversity among those various global populations and historically important subgroups, with the caveat that we only had 3 years to complete the work. And so I think that if wishes were research grants, I would love to expand the reference population and take another look at this question. Now, I show you the same type of plot. So once again, every column is a horse, and we have from our set of 700 plus horses, we selected about 400, if I remember correctly, that were not from families because we wanted to not stack our deck too heavily with one family or another. And we can look at their ancestry both at the wide angle view. So the top plot that is just in green and blue seems to just capture the influence of our modern thoroughbred against all other ancestries. And if we go down to the bottom, to the level of detail that had the best statistical significance, our computer does model the probabilities of each type of view and tells us which one is the most likely. Then we could begin to tease out individual types and subgroups within this global sample of Arabian horses. So I've labeled that for you now. I've kept the thoroughbred in this analysis, and we also included the Icelandic because we wanted to have a nice outgroup to help our computer to distinguish very appropriately the different types and groups within this population. So each of the colors you now see in this rainbow represents a division of that population at some time during history that allowed that ancient or recent, I suppose, that historical population to begin to diverge and develop new and different genetic variations. And right away, you can see some trends in the proportion of each color present in some of these subgroups. The thoroughbred, of course, being pretty easy to pick out in blue there on the right. Here's an example set. This particular type of many different colors is difficult to acquire in a short period of time and is often characteristic of populations that have acquired a lot of diversity over long periods of history and have not yet undergone isolation through a genetic bottleneck, often created by importation, or strong selection to develop a specific body type. That artificial selection from humans also tends to introduce what we call a genetic bottleneck. So this type of pattern of admixture highlights for us, at least among our fairly limited set, the animals that were perhaps older type populations that hadn't yet undergone some of the changes characteristic of our action of modern humans. We also could see within this large block in the center was primarily comprised of horses living in the US and the United Kingdom. And for those pedigrees, it was often difficult to apply a simple human readable label to call them Egyptian or Polish or of a certain group, because these animals, having acquired some ancestry from diverse importation events, often possessed really diverse ancestry documentable in their recent pedigree. So we also see shifts and changes in the number of colors represented in the pattern amongst those colors. Right away, though, you can see on the left-hand side that the horses registered as straight Egyptian have their own type of signal that can be shared in different proportions with the horses that have those diverse pedigrees. Polish horses also, in this case, had a fairly unique signal, again, having gone through an importation to Poland and then being fairly isolated as a result of some of the breeding goals, as well as those periodic political and geographic barriers. They have developed over the last few generations, well, probably tens of generations, their own genetic signal represented here in sort of that lavender color. Now, we can say that, and it's visible in the written pedigrees, that the contribution of each of these subgroups to our modern horses in the United States and in Europe who have those diverse pedigrees is fairly trackable by tracking those shifts in colors. In this case, we could, for this sort of a dusty pink color, identify what looks like a popular sire effect here in the United States, because we could see, at least in written records, commonalities among horses that had a high proportion of that type of pink ancestry. A sample from that particular stallion was not available for testing, so at this point it's just a hypothesis, but it just highlights for you how we have to do a bit of detective work after the fact to go back and figure out what the human readable story might be for each of these biological effects. And then we could fairly well attribute to this light blue type of ancestry. It looked like that corresponded to some horses that derived from our lovely crabbit populations. Now, if we shift to this part of the graph, this is where things get particularly interesting. Here we had some of our older populations, as well as a set of horses that were provided to us with the description that they were racing Arabians, and many of them had racing records or performance records. In this case, we had this unusually strong signal that had the characteristic blue color. And much as it was decipherable in the written pedigrees for the admixture of Egyptian and Polish, in this case, unfortunately, it did not agree with the written pedigrees in most cases. Uniquely, though, we could see some other admixture signals that made historical sense. So in the thoroughbred, of course, we have written records that describe the creation of that breed about 200 years ago using some stallions of Oriental origin. And based on the persistence of some of these red and rust colors in our modern thoroughbred genomes, we believe we can see the signal of that breed creation, that importation of those stallions remaining in the genome of thoroughbreds today. And in fact, we do see this chocolate brown signal that is common in racing Arabians, but also seems to have likely a position in the history of the thoroughbred, and again may reflect, some history of perhaps the Darley or an influential stallion like that. So these populations seem to have influenced one another throughout their history. Now, to investigate this effect further, we used a slightly different technique. In this case, now every row is a horse, and this quilt does provide sort of an overwhelming visual, but hang with me here. So each line is a single horse with their genome displayed from left to right. The color of that particular line corresponds to ancestry, which we have confined the computer to define within just 4 different categories: thoroughbred, Arabians of non-racing origin, the Turkoman horse, and Icelandic as an outgroup. Of course, both Icelandics and Arabians are horses, and so we would expect that if we go back 2,000 to 3,000 years, we would identify shared ancestry. And you can see small portions of green color in each of these lines or rows. What is a bit unusual, though, is the proportion of that green color. As these populations diverge, they begin to lose those shared ancestries. And so much in the way of a molecular clock, you can date when a mixture event occurred based on the proportion of sharing of that ancestry and the size of the individual pieces of that shared ancestry. So when the proportion is small and the length of each of those squares is very small, then we can mathematically calculate that the time since those two horses met one another is very long. And when the proportion is high, the shared ancestry is frequent, and the length of each of those shared blocks is large, then mathematically we can calculate that the time has been very short. In this case, we could identify a set of racing Arabians that had relatively little, in most cases, proportion of thoroughbred genome, which suggested it might be consistent with the known history of shared ancestry between those two breeds in the time scale of hundreds, perhaps thousands of years. But for another set of horses, we did identify a proportion of shared ancestry that was not consistent with ancient mixture events, but instead with more recent admixture events. And indeed, by this measure, we had some horses that were more than half of a thoroughbred ancestral component. And at those degrees, with these long, big blocks of blue, it becomes statistically very unlikely that that dates back more than just a handful of generations or less. Now, the genome also provides us with 2 specific types of nucleic acid that can tell us in a little bit more detail what's happening in the tail male and tail female lineages. And to follow up on that story, we're going to shift now to examine the Y chromosome very specifically. Now, if I make an analogy to an Olympic-sized swimming pool, if we calculated the volume of water present in that pool, it comes out to about 2.5 million liters. So if I compare that swimming pool to the content of the entire genetic sequence within each of the cells, I can then do a bit of math to give you what the volume would be if I was considering only the Y chromosome. In this case, the Y chromosome would compose about 5,000 liters. So compared to 2.5 million liters, there's relatively little information on the Y chromosome. It does have about 37 genes, a small number of which are shared with the X chromosome. So that's about 30 unique genes or so compared to the 20,000 that are in the total genome. So a relatively small impact on the overall biological function with the exception of determination of sex. The unique thing is that, of course, only males have a Y chromosome, and that means it is handed from generation to generation strictly through the male lineage. And so in this example, thoroughbred pedigree, any variation that we find on that Y chromosome, we know with perfect certainty that it must have come from the tail male lineage, at least as long as we're looking at the Y-specific portion of the Y chromosome. Otherwise, when we're looking at ancestry, say the colors I was just describing for those ancestral subgroups, we have to look at it as a population of all of the ancestors as a whole, and we don't have the opportunity to tease apart which piece of that ancestry came from which particular grandparent or great-grandparent. Now, when we apply this same detection of genetic variation to the Y chromosomes in that previous study, we could generate this cluster based on that existing variation on the Y chromosome. This, of course, being work led by our good collaborator, Dr. Barbara Wallner. So in this case, they could create a family tree. And that family tree sort of catalogs the number of differences found between the Y chromosomes. And where the number of differences is large, it's easy to mathematically calculate that the time since those two chromosomes existed in the same animal has been very long. And an important thing to note about genetic variation as it occurs, if every generation we acquire new genetic variations through just random mutation. If one single base out of 2.7 billion changes, it is nearly impossible for that change to reverse itself and revert back to the ancestral state. It's something like lightning striking the same place twice. So we can, with really good certainty, assume that as those variations occur, we can use it to track changes in a single direction because they just don't flip back to that ancestral state. So here, looking at a large population of animals, she could describe Y chromosome variations that very distinctly captured the thoroughbred Y chromosomes, and those are labeled in red and in orange, and then some Arabian type Y chromosomes, which she labeled in green. And then if we examined from that population of horses, males, of course, this won't help us with looking at variation in females, but if we look strictly among males, we found a high proportion of those racing Arabian stallions that possessed the thoroughbred characteristic Y chromosome pattern of variation. So in this case, what admixture is occurring is likely occurring almost strictly through the male lineage because it seems to be very frequent now among those groups. We can also look at this more closely if we compare the Arabian Y chromosome to diverse breeds from around the globe. So in this larger tree created with those 300 plus horses, we can begin to get a much finer detail estimate of the time since those Y chromosomes all existed in one single stallion. So this is it. Imagine this circle as though you're looking at a tree from directly above. And right in the center, we have sort of the trunk of the tree. And then around the edge, we have these many leaves. And each of these leaves corresponds to some modern horses who were genotyped for the project. Now, our colleagues have labeled those leaves based on the number of horses sampled. So in this case, they focused quite a bit on thoroughbreds and Arabians, and you can see the larger circles where they have a large number of horses. And the color also corresponds to their breed. The interesting thing here is that among all of these thoroughbred and Arabian samples, they could date the Arabian-specific Y chromosome back to about 1,000 years between the turn of the millennium and 1,000 years from the current era. So between 1,000 and 2,000 years ago, a very long time. That is a lot of ancestry, a lot of deep, deep history to capture within any single breed. So it's really quite impressive. She also, once again, could see that the blue color found very frequently among thoroughbreds also seems to have found its way into some of our Arabians. So this blue circle here being Arabian horses who possess that thoroughbred type of Y chromosome rather than falling here to the right-hand side with the rest of the Arabians. So again, I mentioned earlier that it's a very old Y chromosome, and it's this divergence, the branching of the tree, that lends to us some certainty that the Y chromosomes that appear to be a thoroughbred origin are not ancestral variation that has somehow been preserved and maintained, but instead a recent occurrence. The probability that it could occur by chance is like having lightning strike many, many times in the same place. It's just not biologically possible for that branch of the tree to have grown backwards into the trunk and then reemerged back out on a different branch. Thing from the study is that they did identify Arabian type Y chromosomes that were no longer found in the Arabian breed. And so these are likely documenting historical use of Arabian stallions to develop other breeds. And in many cases, that ancestral Arabian Y chromosome has actually gone extinct in the modern Arabian and is now only represented in these other breeds. But because of the high degree of similarity, it's easy to track those Y chromosomes to recent common ancestors. So here's an example from the Barbara Wallner paper where the Y chromosome type has helped to provide important information into a bit of a pedigree mystery. So here they were comparing the Y chromosomes that are found in the Arabian family tree, but belong not just to Arabians, but also to Lipizans. And unusually, although if you look at the written records, all of these horses can trace their male lineage back to a single stallion known as Siglavi in the early 1800s. When they look at the Y chromosome types, the Y chromosome did not agree with the written pedigree. And very distinctly, there was a different Y chromosome type found among the Lipizans than among the Arabians. Now, it's possible, of course, that if this difference were due to a single change, that we could say that it was a spontaneous mutation. But in this case, it's due to accumulation of several changes indicating much older history. And the authors propose that this was perhaps a bit of a clerical error because the type identified in the Lipizans appears to be consistent with another stallion contemporary to that era known as Siglavi 4. And so here we have a case where the biological evidence tells a different story from that of the written pedigree. In a much larger study, our friends did go into deeper depth. Here they compared the Y chromosome types from some 1,500 male horses of 189 modern horse breeds. And because they captured that much larger population of 1,500 horses, they could again use that molecular clock approach in counting the accumulation and number of variations to begin to provide a more in-depth and accurate description of key divergences among male lineages in modern horses. So based on the number of variants they identified, they could see evidence of divergence that agrees with human history. In this case, on the Iberian Peninsula, they saw shifts that separated the cold blood ancestors away from the Iberian types of horse that also then were brought to the Americas some 400 years ago. And they could date all of those to some common male ancestors somewhere in the 8th to 16th century. But relevant to us today, they also dated the divergence of what they called this West Asian group of horses to around the 14th to 20th century. So there are some commonalities there that can be seen in the influence of these stallions in modern breeds today. And in this case, we're actually agreeing very much with written history because we do value these lineages, particularly the Arabian and then also the thoroughbred, for specific traits that were then brought into other breeds of horse throughout our development of those groups. Now, of course, we can't forget the other half of parentage, and indeed biology provides for us a similar technology to help to follow the mother's side of the family tree. In this case, we can track not what we call autosomal DNA, so that resides in the nucleus, that blue pillow-like structure inside this graph of a cell, but we're now tracking a very tiny piece of genetic sequence that's found within the mitochondria. So the mitochondria, of course, are the powerhouse of the cell, and they owe their own very ancient lineage to sort of a symbiotic relationship between two single-celled microorganisms. Now, because it once was a free-living microorganism, it had its own genome. And when it became I don't want to call it a parasite, that's not the right word, but when it became an important part of living within another organism, many of the genes contained in the mitochondrial genome were shifted out of the mitochondria itself, where the very dirty work of providing energy for the cell creates an environment that easily damages DNA. But it retained a small piece that we call the mitochondrial DNA for just a handful of genes that can be used to study variation. Uniquely, this mitochondrial DNA very literally forms a circle within the mitochondria, and it's present there in many, many, many copies. That's a little bit different from your nuclear DNA, which is arranged linearly in the nucleus, and you have just one copy of that whole genome sequence. Now, if I compare the mitochondrial DNA to that Olympic-sized swimming pool again, in this case, they're very small indeed. It's about 7 liters of nucleic acid sequence, so very, very small in terms of the total volume if we look at it in single copy. Now, I did just mention that it comes in multiple copies, so but they also hopefully say the same thing. And among that, just over 10,000 base pairs of code, we have 13 coding genes, some very important tools called the transfer RNAs that actually help us to construct all proteins in the cell. And importantly, a section we call the D loop, which is non-coding, which is very advantageous because it accumulates variations at a very predictable rate of change and gives us a nice molecular clock. As you can imagine, creating variation within a gene that makes a protein can be problematic because you can sometimes create a disease and damage the health of the organism. In this case, the non-coding D loop is a very benign region that can freely accumulate variation without risk of damage. Now, the interesting thing about mitochondria is that in an offspring, they're derived almost entirely from the mother, from that egg. This is because sperm need to travel light. They have a distance to go, and while they do need a lot of energy, their mitochondrial genome is considered extra baggage, and upon arrival, is destroyed by the machinery of the egg. I say almost always because there are occasional incidences, and these are extremely rare in biology, where you can find an offspring that has a mitochondrial type that is a little bit different from the mother. And that occurs primarily from two mechanisms: the acquisition of a novel variation, or once in a very, very rare situation, there is some measure of paternal inheritance. One mitochondrion manages to make the trip on the sperm to the egg and is preserved after fertilization. But these types are usually observed as a mixed type because those situations are so rare that they comprise a fairly small number of all those individual mitochondrial genomes in the offspring. Now, because eggs are made only by females, those mitochondrial types are transmitted strictly through the matrilineage. So the offspring will match the mitochondrial type of the mother. That gives us an opportunity to track that tail female lineage uniquely, which is, again, although we have a lot of information about ancestry that can be read from the genome, it gives us a glimpse of the population of ancestors and not always the ability to attribute that information to one single member in that pedigree. In this case, we can track that female membership. So given the power of this approach, several authors have used it to examine the matrilineages present in Arabian horses and compare that to the strains that are often of importance to modern Arabian horse groups. I chose just one paper to bring to you today because it had a fairly large selection of 211 female horses and had labels for those for 13 strains and or modern subgroups from 3 different locations on the globe. In this case, they identified that our mitochondrial or maternal types remained very diverse across geographies. So here, those X chromosomes have been arrayed on a diversity tree, just like the Y chromosomes were, cataloging that variation between individuals. So each of the circles represents a mitochondrial or matrilineal haplotype. And within those circles, what you can see is that the 3 geographies, being Egyptian, what they labeled as desert bred, and Polish, all happen to be shared within virtually every circle. So you can find Egyptian matrilineages coexisting in Polish Arabian populations. So the lineages did not segregate based on geography, which suggests that in each of those importation events, enough mares were used, and those mares had themselves a healthy amount of mitochondrial variation so that even after exportation, they maintained diversity in the mitochondrial haplotypes. They also could label the same tree with the traditional strain nomenclature. And they saw the same patterns of variation. So many horses labeled as coming from a specific matrilineal strain still possessed diverse mitochondrial haplotypes. And in this case, I think we can say that our strains describe and often are written very eloquently in the literature by their type, saying one strain is particularly feminine, one strain is particularly athletic, one strain is known for having larger average body size, and not specifically identifying them as sharing one common female ancestor. That being said, because these strains are used often in historical literature and do seem to describe types, I do think in terms of biology, they are particularly fascinating subjects of future study so that we can better understand how our long history of our human ancestors who treasured and carefully bred these horses have applied their own principles to create the unique and beautiful animal we have today. They did try to model ways to identify those types based on mitochondrial variation, and it was really quite difficult to do. This plot is confusing to look at for good reason. The biologists also found it confusing to look at. So if they attempted to create a computational model to force matrilineal types onto those strains, they could kind of segregate some of that red Polish lineage, but really couldn't do so for all the other matrilineages. And that maybe makes a little bit of sense because remember, of course, that we have had several generations for these mitochondrial haplotypes to begin to separate one another, but on a global scale, those differences are really quite still small. So, thank you very much. I'm trying to leave enough room for questions, but in summary, I think it's important to remember that preservation requires active and evidence-based work, and it is not something that can be left for future generations to do. The effort is needed now and needs to be done systematically and scientifically with the understanding that our human records are not infallible. We certainly can use a lot in genomics to better understand the definition of the Arabian horse today and use that to reconstruct what the Arabian horse of antiquity likely resembled. And alongside of this, I've completely ignored all of the benefits of using genomics to protect health of our modern horse populations, but there are a lot of tools that are very strong for those uses as well. With that, again, thank you so much for your attention. I hope I was able to describe these things in ways that resonate with everyone here today, and I'm happy to do my best to answer any questions.
Thank you very much, Professor Brooks. We have a handful of questions and about 5 minutes. First of all, the high genetic diversity found in those older populations, what does that mean for these populations in terms of their health and also in terms of their value to Western populations with less diversity?
Right. So in many species facing genetic bottlenecks, meaning they've gone through an event, in horses, it's often importation or isolation of a group for a breeding purpose. In wild animals, it's often loss of habitat and approaching extinction. Genetic diversity becomes a concern. When you have less genetic diversity and you have something unfortunate happen in the genome, a mutation that creates damage or a change that is not compatible with life, you often survive that event because you have two copies. And as long as those two copies are different, you frequently are lucky enough that the second copy gives you a functioning protein product to be able to survive. So we see in many species, as the loss of diversity, as there's more similarity between those two copies of the genome within any individual, you see the effects of inbreeding depression. So reduced fertility, reduced body size, and reduced immune resistance to infectious disease, among a number of things. The ability to use genetically documented outcrossing to reintroduce diversity into specific populations is of enormous value in being able to reverse the effects of elevated inbreeding. The great thing here is that if you can identify those ancestries using genomics, sometimes even if there are no written records, you can still, with a very high degree of certainty, define animals that are of particular value and meet your breeding goals, but are not high in those inbreeding values. Hope that answers the question.
Thank you, that does. I also would like to touch very quickly on some of that research that found overlap with thoroughbred genes in racing Arabians. Is it possible to tell which way gene flow went, whether it's from that recorded in documents shared Arabian ancestry or the more recent introgressions, particularly in the case of it possibly being found in the middle of a horse's pedigree? Is there any way to tell there?
Could we identify the specific horse responsible for that introgression? Well, that would be a little bit difficult if you don't have a sample from that specific horse. The Y chromosome is a little bit easier. For that one, you can, in some cases, identify which horse was the origin of the introduction. Now, of course, we do know Arabians and thoroughbreds share history. If we go back many hundreds of years, and you can see that in the Y chromosome, that Arabians and thoroughbreds shared a single Y chromosomal ancestor back around 1,500 years ago or so. But over time, those sections of shared ancestry have become smaller and smaller and smaller. And it's the rate of change in the size of those sections of ancestry that gives us a way to date when that admixture occurred. So by looking at the size of those sections, we can separate out ancient shared legitimate ancestry with modern introgressions that don't seem to agree with the records. It's just a matter of measuring all those sections. Now, that being said, I have asked many times, could I give a percentage of thoroughbred ancestry that would be considered inappropriate in a modern Arabian horse, or would represent an inauthentic pedigree? And the answer is, at this time, because we had limited funding, we did not have a large library of horses. But if we could sample the existing Arabian horses broadly, we could begin to develop a pattern or a range of measures of those shared segments that agrees with those two different types of events, a modern event versus an ancestral one. But as powerful as that tool is, I think that we would want to use it very carefully and conduct some studies to better document what is the existing level of shared ancestry across our modern population so that we can better understand what an appropriate tool might be to help to disentangle these events that may not have been perfectly accurately recorded in the paper records.
Thank you very much. We've now come to the end of our time, unfortunately, but thank you. That was a fascinating and really informative talk. We're very, very grateful to have had you here. Thank you very much.
My pleasure. Thank you for inviting me.